Okay, I hope you're excited because one of the things that people sign up for biology for and really do want to learn about in biology is all the different living things that exist out there. And we're going to go through classifications. And so if you don't remember some of the basics of classification from chapter 16, go review that material. But we're going to be going through the different domains, and within those domains, if there's any different phylums or classes or kingdoms or things that we need to know, that's what we're going to go through. So for this particular unit, one of the things that I have created for students are these comparison tables, and this is a really great way to take notes. So what I would personally do, okay, is I would have these up while I'm watching and listening to the lectures and going through the PowerPoints, and I would be taking notes on the nutrition, location, unique traits, and examples for the different groups I'm going to talk about in microorganisms today in chapter 17. And then I have a similar thing for chapter 18 with plants and fungi, and another one in chapter 19 for animals. This is one of the best ways to take notes and allows you to quickly compare these different organisms. And so I'm going to strongly recommend doing that. And you'll have seen these. These are included in the lecture materials for these particular chapters. So with that being said, let's jump into it. Okay. So microorganisms are not always microscopic. Okay. When we talk about cells and things that are tiny and small, the general rule is that we are going to need a microscope to see them. But just because the majority of organisms require a microscope does not mean that they are all microscopic. However, when I look at the whole, a really easy way to classify things like viruses, bacteria, archaea, and protists are as microorganisms. They are generally going to require a microscope to see them, and so we kind of clump them together. If you're curious about any of these groups as a whole, I strongly suggest taking any sort of microbiology class that you can take because you do spend a lot more time talking about all of these different groups within a microbiology class. So this is kind of a crash course microbiology. So here are our objectives. We're going to define and describe viruses. We're going to talk about viral reproduction, which is really kind of replication. And so, some places call it reproduction. I prefer to use the term replication, but your book does call it reproduction. We're going to go through prokaryotic cells. So viruses don't have cells. They're not alive because they do not have the 6 characteristics of life. But then we're going to talk about prokaryotes, which, if you remember, do not have a nucleus, and that includes bacteria and archaea. And then finally, we're going to talk about eukaryotes called protists who do have a nucleus, and these are going to be your little pond critters, generally speaking, and we're going to talk about those a little bit too. So how do we classify these groups? What do we do? Let's start with viruses. Okay, viruses, again, are non-living because they are non-cellular. They do not have cells. Generally speaking, viruses are much, much smaller than any other microorganism we would look at, but there have been giant viruses that have been found that are larger than bacterial cells. So it's, again, if you hear all or always or never in science, you should be skeptical because that's not going to be 100% the case. There are exceptions to most every rule, okay? A lot of scientists do argue because they have a lot of characteristics of life that they should be considered living, but based on the current characteristic parameters, they are not alive. They are obligate intracellular parasites, which is a very fancy way of saying that they need to be inside of a host cell in order to go through their replicative process. So outside of a host cell, viruses are called virions or viral particles. And they are basically just a protein capsid, so a package of protein with DNA or RNA inside in very, very tiny, small genomes. That's it. So they don't have organelles. They don't even make their own lipid bilayer. That would be something they might take from a host cell. They are really, really simple when we compare them to our other complex cellular organisms. Some viruses have spikes for attachment. That's the classic, the COVID pictures. They always have their little spikes. And some cells have an outer envelope like the flu, and it's considered an enveloped virus, and the envelope allows the virus to survive longer outside of a host cell. So it can be really beneficial to that particular virus. Most viruses don't survive long out of a host and do require some sort of direct transmission from an infected individual to a healthy individual. Okay. When we talk about viral reproduction, or as I like to say, replication, because it is more like a copying machine, they are just kind of making copies. Put in a piece of paper, you hit 50 copies, you hit start, and you get the 50 copies of the same paper. That's really what we see with viruses. But they are very, very host and host tissue specific. So the viruses that infect bacterial cells, like the bacteriophage, is not going to, like, I have a little bacteriophage. Sorry, I remembered. Look at it. It's a little 3D printed bacteriophage. This bacteria is the very classic alien T4 bacteriophage, cannot infect human cells. It does not recognize human cells as a potential host, and so there is no infective aspect of it. Viruses are host cell specific. So you don't get cold infections on your fingers, you get cold in your mucous membrane and respiratory tract because cold viruses can only impact those particular cells. Once they find a cell that they can infect, they can interact with, they do recognize as a potential space, they will inject or insert their genetic material. That viral genome is then going to go through one of two replicative processes. Okay, let's talk about the first process, which is the lytic cycle. We remember, hopefully, that lytic or lyso means to break or burst. So both of these cycles will end up with the host cell breaking or bursting and becoming damaged and unalive. Okay, but if a viral particle attaches to a host cell and injects its DNA or RNA, its genetic material, and that genetic material is used to very quickly generate new viral components that are then assembled together in an assembly line, and then that virus leaves the host cell by bursting or budding. That is the lytic cycle. This is generally really quick, acute onset symptom viral diseases, so things like cold, flu, norovirus. Norovirus is a stomach flu, which causes vomiting and diarrhea. Generally, if you get an infection, you're going to have symptoms fairly quickly. Like, you could be exposed to a cold and have symptoms within like 2 days. COVID and coronaviruses follow this. And so when we see, a lot of viral diseases follow the lytic cycle. Okay. But not all. Some viral diseases have the ability to insert themselves into the host's genome. In doing that, it switches from what would be a lytic cycle to a lysogenic cycle. The lysogenic cycle, lyso meaning burst, and genes means that they spend time in the genes and they burst later. So this second cycle is, again, the viral DNA or RNA enters the host, and then it is integrated into the host's genome. Once it's in there, every time that host cell goes through mitosis and makes a copy of itself, the genetic material from the viruses are copied to the offspring. So you can lay low, hang out under the radar as a virus, and get pushed out to a bunch of new cells until such a time that there's some sort of signal. Usually it's some sort of stress signal that tells the virus that, ooh, this host is not doing so hot, time to find a new one. And we'll see those viral cells then switch back to the lytic cycle in the part of generating the viral particles and then leaving the host cell by bursting or budding. Lysogenic cycle is something we see in humans. HIV is a big one. HIV is going to integrate itself into the T cells of the immune system, and as those cells replicate, we do see that they become more and more infected with the HIV. When that HIV virus becomes activated and starts bursting and leaving those T cells is when HIV becomes AIDS, and you see that immune deficiency. Okay. Another really common one is chickenpox. So chickenpox, if you get it as a child because you didn't have the vaccination, like I did, we had pox parties. Okay, kids got sick. You took your kids to get the chickenpox. It's much safer to do the vaccination. The chickenpox, the initial outbreak is a lytic cycle. You're going to have those itchy sores. Those sores are shedding live virus, and so it is going through that lytic cycle. But not all chickenpox virus will go through the lytic cycle. Some of it will go into a lysogenic stage, and then as an adult can reemerge as a different disease called shingles. We see that in the lysogenic cycle as well. So when we talk about viral replication, there's many different ways that this can happen, but those are the two big ones. And I do want you to be able to tell me the difference between both, okay, with examples. Just gave you some examples, all right? Here's our first check your knowledge, okay? Pretty, I think, straightforward. If you can answer those questions, you're good to go. And let's go to the next part, which is going to be bacteria. One of the common misconceptions is that if you have a cold, you need to go get an antibiotic. And that started becoming a thing in the 1940s and 1950s when antibiotic use was discovered and increased in human populations. Viruses are not affected by antibiotics. Antibiotics are only going to be effective against bacteria. And bacteria, not all of them cause disease. So if you do get sick and it seems like it's resolving itself pretty quickly, it's probably virus. Or if you go to the doctor and they're like, listen, it's viral, you have to just kind of suck it up. That's because if they give you antibiotics, you're more likely to have other issues. Okay, so there's my little caveat about antibiotics and viruses and the difference there. When we talk about bacteria, these are prokaryotic cells, so they do not have a nucleus. They have that nucleoid region, which is still very organized and maintained. And these are small prokaryotes. They're generally much smaller than eukaryotic cells. There is a giant bacteria that is bigger than a fruit fly, which is wild to me. Absolutely fascinating. They don't have membrane-bound organelles, but they do have some things that we can use to identify bacteria and classify them as a bacteria. The first being that they have three generally common structures: rods, spheres, and spirals. Aside from them being prokaryotes, that's number one, prokaryotes, okay? Rods are bacilli, or rod-shaped, kind of oval-shaped. Spheres are going to be circles. These are often called cocci when we're talking about it in scientific terms. And then spirals, or spirula, or spirochetes, and they look just like spirals, okay? Bacteria reproduce asexually through binary fission, which is a kind of mitosis. I have that in picture B here. You can see that pinching of that cytoplasm and that division into two cells. And then the outer cell wall is strengthened by something called peptidoglycan. Now, not all bacteria cells have peptidoglycan, but a lot of them do. And that peptidoglycan can be a very useful differential tool if we're trying to determine if it's something that's bacterial and what kind of bacteria. Okay. Now, bacteria are something we call ubiquitous, meaning that bacteria are found literally everywhere. Literally everywhere. You have more bacterial cells in your body than you have human cells. And a lot of doctors have shifted to treating your microbiome like its own organ system, because your microbiome can have such huge impacts on your health and mental well-being that we need to treat it with appropriate care. All right. So how else do we classify bacteria? How else do we identify them? Well, bacteria are going to have, because they're found everywhere, they're found in all sorts of different environments, and we're going to see bacteria having really wide nutritional options. There are three classes that I'm going to ask you about. They're not the only ones, okay? If you take microbiology and you're like, well, Ms. Larson only taught me these three, please know that you're going to learn more, okay? But we have photoautotrophs. So these are light self-feeders, and these are your blue-green algae. They used to be called blue-green algae, but they've been reclassified as bacteria, and that's why they are called cyanobacter or cyanobacteria. Cyanobacter is actually super cool. I love cyanobacter. I'm pretty sure this one in this picture is one called Anabaena. Oh, it's so good. Cyanobacter is one of the first organisms to start producing excessive amounts of oxygen on this planet and are likely the cause of something called the Great Oxygenation, and the reason that we use oxygen as our electronic scepter. I know some of you have asked potentially about that in your attendance quizzes, so if you're wondering, yes, Cyanobacter, awesome, awesome guy. There are a lot of different ways that bacteria can utilize solar energy and carbon dioxide to make food. There's a lot of different varieties there. We do often find them in wet aquatic environments because they are in a solution of nutrients and water. There are also chemoautotrophs, which are not using solar energy or carbon dioxide, potentially, to go through the process of making their own sugars. So at the bottom of the ocean, around these hot geothermal vents, there are things called tube worms. Tube worms get to be very, very large, and they have a symbiotic bacteria that is using the geothermal energy to produce sugars. And that's how the tube worms stay alive, and the bacteria do well in the tube worms. And so that's how we see that. We're going to find bacteria everywhere, and so we will find some organisms who are making their own food without utilizing solar energy. We also have organisms called chemoheterotrophs. This is similar to us. We take inorganic molecules as our source of energy and carbon. And so this is something that we see when it's other organic feeder. So chemoheterotroph, they're eating something else. One of my personal favorites are the saprotrophs. Saprotrophs are the ones that are responsible for decomposition. When you see like rotting, if it's not a mold or a fungus, it's a bacterial that's rotting, and it sends enzymes into the environment to digest food. A lot of saprotrophs potentially have antibiotics as part of those enzymes because the soil world is a battlefield, and so we do see those guys playing a role in medicine as well. So, what about bacteria in the environment use you? Again, I just mentioned that bacteria are important decomposers. Without decomposers, bacteria or even the detritivores like your worms and your vultures or your fungi, we would be in really big trouble. Life would halt without decomposers because that nutrient cycle would be disrupted. Carbon would not be available from dead and decomposing material because it wouldn't be decomposing material. Okay? In addition, there are mutualistic bacteria that live inside root nodules and other plants. If you've ever pulled up like a pea plant or a bean plant and you saw these little bumps on the roots, I don't know, maybe you're not a weird kid like I was, but I used to do this. And those roots are going to have bacteria in them that are capable of trapping nitrogen for the plant. So that's really, really beneficial to the plant. And for ourselves, we have a lot of mutualistic and even commensalistic bacteria that live in our microbiome, on our skin, in our mouths, in our guts, in any area that we have that is exposed to the world. And we do know that those bacteria play a really good role in your health and well-being. There's been a lot of research that is suggesting that your gut microbiome could have impacts on mental wellness, as well as obesity and risk of diabetes and other diseases as well, IBS, Crohn's disease, that sort of thing. So, they're saying like 90% of diseases can be reached in some way back to the gut and the health of the microbiome. So, if I'm not saying go take a bunch of supplements and probiotics, but they can be helpful, especially if you're on antibiotics. Eat some yogurt and that good fermented food. But in addition, just, eat some fiber. I keep saying it, but your gut microbiome needs fiber. So, feed your microbes. When you're eating good, healthy, balanced food, it's because you're taking care of your little bacteria pets. Maybe that'll help you avoid the McDonald's for the third time today. Okay? Guilty. I've been there. I'm guilty of it too sometimes. All right. Bacteria can be really beneficial outside of our bodies. We have a lot of ways that we use as humans bacteria to help in our environment. We call a lot of this bioremediation. So bacteria can break down pollutants that exist in the environment, and we've been exploiting those, including there are bacteria that are capable of breaking down microplastics and other plastics, which is hugely important because plastics have become a big problem. If we talk about, why that is, well, if plastic is a food source that nobody else can eat, you now have access to an all-you-can-eat buffet where there's no competition. So there is a natural selection and pressure for those bacteria to be able to consume those plastics. We can then find some of those bacteria that are beneficial, and we can bioengineer them to be even more efficient. And so we have times where, like the Deepwater Horizon spill in the Gulf of Mexico, we've gone through and we've added bacteria that can break down oils to help speed up the recovery from that major catastrophic environmental factor and event. So, disaster, really, if you think about it. That's what bioremediation can be used for. On a regular, everyday level, bioremediation is used in wastewater treatment, or if you have a septic tank, you can add things like RidX, which is a brand of bioremediation. You're adding bacteria that are going to break down those solids in a sewage tank and a septic tank. That's going to allow that septic tank to last longer between needing pumped. So huge benefits there outside of the human body. And again, bacteria are huge parts of fermentation. And if you like fermented foods, you like bacteria. Fermentation produces that lactic acid. So if you like those tangy yogurts or sour creams, that's that tangy flavor, and that's coming from those bacteria. It can pickle cucumbers. It can turn milk into cheese. I love cheese. I'm from Wisconsin. Okay. I actually worked in a cheese store. So, like, I don't think I can get away with not liking cheese. Most antibiotics were discovered from soil bacteria because, again, it's a battlefield for resources. And antibiotics are a naturally selected factor that allows individuals to survive better than their neighbors by killing their neighbors and taking their nutrients. So, bacteria is huge in there. We are actually using bacteria to bioengineer different medicines like insulin. Insulin used to be made from the pancreas of baby cows, and then it was made in horses and their blood. And you can imagine that both of those are both ethically questionable for a lot of people, as well as expensive and prohibitively expensive. Insulin nowadays is mostly being made by a strain of E. coli. And I don't know of anyone who gets upset when we put E. coli in the autoclave and kill those bacterial cells. It's also why I don't think insulin should be as much as it is. And I think most people would agree with that. My mom is on insulin, and I hate that it costs her so much because she doesn't want to take it. And it's a life-saving medication. Vaccines are often made through bacteria being bioengineered, human growth hormones for people who are in need of that. There's a lot of really good things happening there. Okay. Bacterial diseases. This is when you're probably going, but Ms. Larson, bacteria makes us sick, right? Bacteria make us sick. They're the germs. They're the germs. They're the germs, right? 0.1% of known bacteria are known to cause human illness. That's it. That's it. Of all the bacteria that we know. Now, mind you, there are some we haven't discovered yet and things that haven't been described, but only 0.1% are known to cause illness. Now, I'm not saying those illnesses should be overlooked. They can be really bad. Bacterial meningitis is caused by a whole list of bacteria that can cross the blood-brain barrier, and any sort of swelling in the brain is really of concern and life-threatening. But you can get, like the sexually transmitted diseases, pneumonia, skin infections, things like Staphylococcus aureus, though. You'll hear about like Staph aureus, MRSA, right? Methicillin-resistant Staphylococcus aureus. Staph aureus is a normal skin bacteria. It is found on a lot of people. It is something that can become an opportunistic pathogen or take advantage of someone being ill. So when we talk about a lot of these bacterial diseases in humans, most of them are going to be bacteria that are normally colonizing human individuals, and they just become problematic when systems are disrupted, which is why your microbiome as a whole can be really important, okay? I'm not saying you can't get like salmonella from eating raw eggs that you didn't wash. Absolutely. But if I look at salmonella then on reptiles, it's a commensalistic or even potentially mutualistic organism, and it is not something that's causing disease in reptiles like birds. So there you go. Okay. All right. That's it for bacteria. Hopefully that answered some questions. Again, this is a very, very brief overview compared to what you would get in a microbiology class. But those are your bacteria. I'm going to very, very, very briefly talk about archaea. So bacteria are prokaryotic cells, and for a long time we classified archaea as a type of bacteria. But in the 1970s we determined that they actually share ribosomes with eukaryotes, not bacteria. And so these are prokaryotes that are related to eukaryotes, okay? They have plasma membranes with really unusual lipids, and so as such, archaea are often considered extremophiles, and they love living in extreme environments. If we compare archaea to bacteria, they don't really have common shapes. These are some common archaea in this picture here. They don't have the cell wall made of peptidoglycan like we see in the bacteria. While they do share some nutrition, these guys are really, really unique, and there isn't a whole lot known about them. It's a very big area of research right now, but one of the things we do know is that there aren't any archaea that are known to cause human illness as of right now. Okay, let's talk about the different kinds of extremophiles that exist. So in our extremophiles, we have a lot of different ones. I'm going to tell you about 3, and I want you to just know about them, okay? So Archaea can be found in methane environments. This includes swamps and animal intestines like cows and termites, and they are going to break down cellulose and other materials into methane. So we call them methanogens, and it's the reason why cows burp and fart a lot. Okay. I personally really, really like halophiles or halophiles because they live in incredibly high salt concentrations and they actually require a lot of salt, which we know that a lot of salt would generally result in a cell being in a hypertonic environment and losing its water. These are adapted to handle that. In fact, they need it. So if we go to like the Dead Sea, some of those saltiest places on Earth, we will find archaea, but we won't find most anything else alive there. And then last but not least, and definitely not actually last if we look at archaea as a whole, we have thermoacidophiles. So these are organisms that survive in extremely hot acidic environments, like the thermal geysers at Yellowstone National Park. I don't know if you ever follow some of the national park services and the what people call tourons, the tourist morons at these national parks, but the areas around the geysers at Yellowstone are incredibly hot and acidic. There was a gentleman several years ago, and I've double checked this to make sure I'm not crazy, who fell into one when he and his sister were trying to find a place that they could hot pot, which is like skinny dipping in a thermal acidophile. I don't know. They were going to do that. He fell into one, and by the time they got back to help him, it was about 24 hours later, and they only found bones. He had boiled to death, and then the flesh on his skin had dissolved. That's really a harsh environment. There was a bison in 2025 that walked into one and died in front of tourists because it is not an environment that these guys can live in. But these little archaea, tiny little cells, doing a great job, living their best life. Okay, that's all I really have on archaea. So make sure you look through your prokaryotes, bacteria and archaea, as well as your viruses. And then we're going to talk about eukaryotes, microscopic eukaryotes. Okay, all right, ready? Protists. Protists are awesome. Protists are the junk drawer of eukaryotes. So they all have a nucleus. They all have membrane-bound organelles, but we don't really know where they belong. So it's what we call the catch-all, and it's a classification of not fitting somewhere else. So if the organism is not a plant, an animal, or a fungus, it is considered a protist. As such, they do not have a common ancestor of all protists, and this is meaning like this group is not really well classified, because when we talk about classifications, we're saying here was one common ancestor that split into these different groups. We don't really have that for protists, and so they might be several super kingdoms or superphylum. We don't really have a good classification. It's a classification of exclusion. That being said, they are eukaryotes, and they do generally tend to be unicellular, but not all of them. And they do generally tend to live in aquatic environments, but not all of them. So we're going to talk about a couple. We're not talking about all of these different groups today, but what I'd like you to note is that fungi and animals share a common ancestor, and there are a couple of different groups of protists that might be related to that. And then algae is a protist, but it is a sister group to terrestrial land plants. So that's why it's plants and algae when we talk about things like photosynthesis. It's not just the plants, and that includes algae. They're separate. So, protists. Protists are often classified by their nutrition and movement. So again, not a plant, animal, or a fungus, okay? And then we look at other characteristics. We go, okay, they're generally unicellular, they're generally aquatic. We can also generally identify groupings by energy and nutrients. So we have individuals who can be heterotrophic or other feeders, autotrophic or self-feeders, or even things that can be mixotrophic, which is where they do both. It's a very classy name, right? We're like, oh, they do both. It's mixotrophic. Now, there can be really big benefits to all of these. So individuals who are heterotrophic obviously can eat them other organisms or dead and decomposing material, and a lot of them do play a role as decomposers. Things like paramecium, some of them can photosynthesize, and so if they have plenty of sunshine, they can go through that process of photosynthesis. And then you have euglena, which does both. So Euglena is capable of both photosynthesizing and eating other things. And honestly, if you live in a really variable world, like one of those retention ponds that are out here in North Carolina, where it's like hot one day, cold another, sometimes it's sunny, sometimes it's raining, it's really lots of water or not a lot of water, being able to be variable and versatile in your feeding can be really beneficial. Okay. We also see a lot of diverse movement in these groups. We can see movement by flagella, such as our euglena I have in the picture here. It has a little flagellum and it kind of spins around. The flagella moves and the euglena kind of spin. It's pretty cute. You can have cilia, which move in a oar-like motion. They're hairs on the outside of the cell that move in an oar-like motion. Or my personal favorite, pseudopods. Pseudopods move by cytoplasmic streaming, and I love watching amoebas move. They push out these little fake feet, and then they kind of pull themselves in that direction, and it's mesmerizing. If you're wondering about any of these protists, these little aquatic ones, you should check out Journey to the Microcosmos with Hank Green and James Weiss. Wonderful series on YouTube, just exploring pond water because pond water is awesome. Okay, the scummier the better. Let's talk about a couple different groups here. I'm not going to talk about all of them. There's a lot of different protists, but there's some that we see commonly that we're familiar with, maybe because they cause disease or maybe because they're just relatively common. So the protozoans are a very big group, and a lot of them can cause human illness. So these guys reproduce asexually. They usually have complex cells, but they are unicellular, and they're most often heterotrophic and move in some way, shape, or form. I have 4 examples on this slide here. You have your ciliates, okay, they're going to move by that cilia motion, things like paramecium. Paramecium are very, very common in pond water. If you ever get a microscope and you're looking at a sample of pond water through it and you see these little guys beep-bopping faster than you can get a bead on, those are likely paramecium. Okay, amoebas move by using those pseudopods, and they're amoeboids or amoeba-like creatures that include your slime molds. Amoebas are really interesting because they do cause some human illness. They can cause amoebic dysentery if you ingest them and they get into your intestinal tract. And then in 2025, there were actually a lot of cases, like probably more cases than I think there had been in recent years, of that brain-eating amoeba. Now, I know people hear brain-eating amoeba and get scared. It's a particular kind of amoeba called Neisseria fowleri, and it lives in really warm, still water. And if you accidentally are swimming, you get water up your nose, the brain barrier between your nose and your brain is very, very thin, and these guys can slip between cell junctions and make their way into your brain. So it is something to be concerned about, especially if you're out enjoying water sports and it's really warm water that's really not moving a lot and you see a lot of waterfowl, because Neisseria fowleri is a amoeba that is found in the intestines of ducks and geese. Okay. The next picture is Giardia, which causes giardiasis, and it's kind of a zooflagellate. So Giardia is, if you ever see people in like Naked and Afraid or Alone, and they drink water without boiling it, and they get really sick, a good chance is that they have Giardia. Giardia gets into the intestinal tract and will cause really severe diarrhea and recurrent infection. So it doesn't just clear up. It feels like it clears up, and then it comes back like 2 weeks later. So that's a problem. If you have a dog who's a puddle licker, get your dog vaccinated against Giardia, because if they're a puddle licker and then they're having bouts of explosive diarrhea, they probably have a Giardia infection. Okay, my parents had a dog that was a puddle licker, and I kept telling them to get him vaccinated. Okay. Apicomplexans don't move, but they are very problematic in humans. This last picture here is an example of malaria. Malaria is a kind of protist that does get into red blood cells and can cause death and illness in humans, and it is still one of the leading causes of human illness and death in this world. So that is a protozoan, not a bacteria. So if you've ever wondered about that, there you go. Then we have the algae. I love algae. Algae and cyanobacter produce 50% of the oxygen as well as phytoplankton on this planet. So we think about, oxygen production and photosynthesis. We need to really appreciate algae. Now, algae, if you have a fish tank, can be a problem, but you can get fish that eat them. These generally have a cell wall made of cellulose, but they don't have roots and shoots. So they don't have roots and leaves. They do have chloroplasts, and they are similar to plants in that way. And so we see a common ancestry between plants and algae. Some of them can move and have flagella that move them around. Some of them cannot. Some of them have things called holdfasts, which allows them to anchor to a plant. Some of them are free floating. There's a lot of variety within the algae themselves. Some of them are very tiny and microscopic, and then others are entire ecosystems like the kelp forests off the coast of California. So, algae are going to be one of those classic protists that are incredibly variable, but none of them are plants. They are commonly related to plants with a common ancestor, but they are not plants themselves. Um, I love algae, and if you ever eat seaweed or nori, you are eating a protist, you're eating algae. Okay? And then my personal favorite are the slime and water molds. I just think these guys are so fantastic. Slime molds are common here in North Carolina. This is actually a picture of a slime mold on the top that I took in my neighborhood on a tree, and it was about the size of a dinner plate. I was taking a walk, and I was like, I just slammed on the brakes. This is one of my favorites. This is called Plasmodium, or the many-headed slime mold, also known as dog vomit slime mold, also known as scrambled egg slime mold. It's fantastic. And I see it in North Carolina here a lot because we have very damp environments, and they do help recycle dead plant material. And they feed on bacteria and they feed on other fungi. But so they were classified as a fungus for a long time, but they move like amoebas and they have been genetically shown to not be fungi. They are actually a protist. And then water molds decompose remains in aquatic environments, which is still important. We need decomposition to happen in water environments for when plants and animals die in an aquatic environment. Water molds can also be really significant parasites. So this is a fish that's infected with a water mold. It's not ick. Ick is a protozoan, so ick is another protist. If you have a fish tank and you ever gotten fish that had ick, this is a different kind of thing. It's a mold, and we actually think, I'm pretty sure this has been shown and supported, that the potato blight of 19th century Ireland was caused by a water mold, and that changed the course of history. Arguably speaking, on a lot of different levels. So just a fun little fact there. Protists are their own really fascinating area of study. I don't expect you to know all of them, but I do want you to know these couple here. That's it for today's lecture. Do check your understanding on your protists. Let me know if you have any questions and fill out your table. Have a good day.